Assalamu alaikum. This is Ahmed Shams. Staphylococcus aureus. I think it's the most famous bacteria in the world. And beta lactam antibiotics. I think they are the most popular antibiotics in the world. There is a historical sequence of events in resistance of Staphylococcus aureus to beta lactam antibiotics. But history is not our goal in this lecture. Instead, we are going to arrange our scientific knowledge in a chronological way. Before we show how Staph aureus was transformed from a peaceful bacteria, almost peaceful, into an evil bacteria resistant to almost all beta lactams, we should see how Staph aureus resists beta lactams. I mean the mechanisms of resistance. The first mechanism is destructing beta lactam by beta lactamases. The second one is by target substitution, and I want to stress on the word substitution. It's a target substitution, not a target modification. I mean, when Staph aureus gain this type of resistance, it doesn't modify the target site, the penicillin binding proteins, which beta lactams attach to. It produces a different type of penicillin binding proteins. So they are alternative penicillin binding proteins. In other words, it's a surrogate penicillin binding proteins. It's a surrogate target, not a target that was modified to resist the antibiotic, but it's another target. I like this slide. That's why I made a separate video of this slide. It's on my channel on microbiology playlist. This slide shows how MSSA, methicillin sensitive Staph aureus, is transformed into MRSA, methicillin resistant Staph aureus, by gaining a gene called MIC-A. And I'm sorry, there is a mistake. MIC-A gene, the M should have been written in a small letter. Anyway, Staph aureus normally has about 4 types of penicillin binding proteins, normal penicillin binding proteins from 1 to 4. These penicillin binding, the most essential one is penicillin binding protein 2. These 4 types of penicillin binding proteins are sensitive to beta lactam ring. When on the Staph aureus, or when these penicillin binding proteins from 1 to 4 are subjected to a beta lactam ring, they are inactivated. But when Staph aureus gains MIC-A gene on what's called staphylococcal chromosomal cassette, which is a transmissible genetic element, when it gains this gene, MIC-A gene, this gene produces and synthesizes a penicillin binding protein 2A, which is resistant to beta lactam ring, because its active site serine, the red ring or red dot, is buried deep in the cleft. So the beta lactam ring cannot reach this active site serine and cannot inactivate this penicillin binding protein 2A. If this penicillin binding protein is not deactivated while other penicillin binding proteins from 1 to 4 are inactivated, so it can complete their function and can replace their function. It can keep peptidoglycan layer intact because it's responsible for cross-linking peptide together in the peptidoglycan layer, and can keep the Staph aureus cell wall intact. So in this way, Staph aureus can survive beta lactam antibiotics, and it's called MRSA, methicillin resistant Staph aureus, because methicillin is a representative of penicillinase resistant penicillins or penicillinase resistant beta lactams. Now it's time to tell you the story of Staph aureus resistance to beta lactams. This story, or the first round of this story, started in 1942 by the introduction of penicillin in clinical use. At first, penicillin could defeat Staph aureus, but within a few months in the same year, Staph aureus responded by production of a penicillinase coded by a gene named BLA-Z gene. So it was a short time victory. That penicillinase is a narrow spectrum beta lactamase. It can deactivate penicillin and aminopenicillins only. Aminopenicillins are amoxicillin and ampicillin. Rarely, Staph aureus containing that BLA-Z gene can hyperproduce that narrow spectrum penicillinase. In that case, this narrow spectrum penicillinase is no longer narrow spectrum. Its spectrum will be broader, and it will gain the power to deactivate stronger penicillins. These stronger penicillins are called penicillinase resistant penicillins, and though they are penicillinase resistant, but they cannot resist hyperproduction of penicillinase. So this narrow spectrum that's hyperproduced becomes broad spectrum, and it can destroy methicillin, oxacillin, and cloxacillin. And these rare strains are called BORSA. Borderline oxacillin resistant Staph aureus. It means they can resist oxacillin without the presence of MIC-A gene. They resist oxacillin by hyperproduction of penicillinase. Round 2 of the battle. From the year penicillin was introduced, 1942, to 1959, when methicillin was introduced, Staphylococcus aureus kept producing narrow-spectrum penicillinases, and we called it methicillin-sensitive Staph aureus. On the introduction of methicillin, narrow-spectrum penicillinases were defeated, but this victory of methicillin lasted for 2 years only, because in 1961, Staphylococcus aureus gained the power to destroy methicillin, and it was called methicillin-resistant Staph aureus. And as you see, it gained that power by gaining the MIC-A gene. When we speak about that gene, which is carried on a staphylococcal chromosomal cassette. And in this drawing, you can see that the MSSA gained the big red spike or red horn MIC-A gene. And to remember that most of the MRSA still produce penicillinase, so they have the BLA-Z gene. Round 3 of the war, and this round of the battle or the war is still going on in our time. From 1960 to 2010, there was no beta-lactam antibiotic able to defeat MRSA. In 2010, a fifth generation cephalosporin named ceftaroline and its brother ceftobiprole were introduced in the market. They could defeat Staph aureus or MRSA in particular because they could reach the active site serine of penicillin binding protein 2A, which is coded by the MIC-A gene. They can reach the active site serine in the cleft, in the deep cleft, and inactivate this penicillin binding protein 2A. But there is no happy end in this war, and Staph aureus could fight back. Staph aureus fought back in 2 ways. If it was MRSA, it mutated its MIC-A gene. So this gene that was acquired, it could mutate it. So it resisted the ceftaroline or fifth generation cephalosporin. And if it is MSSA without a MIC-A gene, it could point mutate its penicillin binding protein 4. And although it was thought that penicillin binding protein 4 is not important in Staph aureus, recent studies show that mutation in penicillin binding protein 4 made Staph aureus very resistant to beta lactams, especially the 5th generation. This is the end of this fight until now, and see you in the next lectures.